Hello everyone, my name is Tsu-Wei Chen. Welcome to my presentation. In this presentation, I will show you an ongoing project where we use the greenhouse experiment and also phenotyping platform in combination with the field data to understand the functions of phenotypic plasticity in plant-plant interaction and the canopy productivity. First of all, plant-plant interaction can occur in any canopy, and it can be competition or cooperation. In case of plant-plant competition, it is possible that the canopy productivity will be decreased. Plant-plant competition can occur from the homogeneous canopy like in the farmer's farm. In this case, this kind of competition is the intragenotypic competition. With the increase in canopy complexity, it can also be intergenotypic competition that typically is in the breeders farm in the F2 to F4 generations. In this case, each neighbor plant belongs to the different genotype. The question now is how this intragenotypic competition and the intergenotypic competition affect the canopy and the individual fitness. To understand the role of plant-plant interactions on the individual and canopy productivity, we first start with an experiment with 3 different treatments. T1 is that the plant with individual grown individually with the least competition, and T2 is the treatment with shoot competition but without root competition by separating the root zone of each individual plant. And T3 is the competition with the shoot and also the root. And in this experiment, we use 223 genotypes that represent the breeding history of German winter wheat. And with these 3 treatments and 4 repetitions with the 3 harvests. And preliminary results already showed that the competition reduced the individual and also the canopy productivity. By comparing the shoot dry mass between T1 and T2, we clearly see that there is no clear relationship between their dry mass. That means the competitiveness, so to speak, the dry mass in T2 and the plant vigorousness that is represented in the dry mass in T1 are completely different traits. And then the increasing the degree of competition, that means the comparing the T2 and T3, we also see that the plants in T3 has lower dry mass. That means generally competition reduces not only the individual fitness but also the whole canopy productivity. By plotting the shoot dry mass with the year of release of all cultivars in 3 treatments, it is surprising to see that under the high competitive environment, let's say in T2 and T3, modern cultivars produce less shoot dry mass. This means that modern cultivars are less competitive in comparison with the old cultivar. And this could also an indication that the breeding success is probably achieved by promoting plant-plant cooperation in a homogeneous canopy. To prove that, we conducted the second experiment using 200 genotypes with 3 different plant density with the same genotype. That means this is an experiment of intragenotypic competitions. This experiment was conducted in Inha Montpellier in the PhenoArch platform. And then every day we get the images of the plants that allowed us to reconstruct the 3D architecture of individual plants. We are going to use this data to construct the 2D morphological descriptor and also the statistical model of the trait estimation. And furthermore, we can use the binarized the image data to reconstruct the 3D architecture and to get the 3D morphological descriptor, and also use the 3D architecture model to simulate the light interception of individual leaf in the simple canopy or also in the complete canopy, and to quantify the degree of cell shading and also the competition and competitiveness of individual plants and to compare different genotypes. Here I will just show you an example from two genotypes, the genotype 222 and the genotype 88. You can clearly see that under one plant conditions and also 9 plant conditions, already plasticity in plant structure can be already observed. And we are going to explore this plasticity further in details. To summarize, the project is just starting since last year, and now we have got already 2 data sets with the homogeneous canopy and also heterogeneous canopy. And then we are going to use the 3D plant to conduct the functional structural plant modeling approach and to understand how the phenotypic plasticity has affected the breeding success in the last 50 years, and how can we use this knowledge to further improve the breeding success in the future. If you are interested in our work, please you can follow us on Twitter. Finally, I would like to thank the team in Berlin and also the team in Montpellier that allowed us to conduct these 2 huge experiments. And of course, I also thank the DFG for funding me the Immunity program and also the European Plant Phenotyping Network EPPN 2020 that found the experiment in Montpellier. Thank you for your attention.